So I'm just going to move on to my actual role, which is in terms of genetics and understanding. And I wanted to put our kind of point of view across ultimately. So I know everybody here will be familiar with the wording on the post-mortem form, and this is taken from one of the local teams, and I think it's fairly similar in most places. And I think what comes out and what is very obvious when I speak to people generally is that genetic testing is this terminology that, what does it actually mean? It could mean anything. And so when people say, oh, they've had their genetics done, as a geneticist, that doesn't mean very much to me, because I want to know what type of testing they've had. So I just wanted to clarify a couple of bits about what genetic testing is and what it may consist of. So this is just a little brief bit of revision for anybody. It's probably quite a long time since people have looked at science and things like that. So what we're talking about when we're talking about a carrier type is looking at the actual chromosomes themselves. So that's like a big overview of the person's genetic makeup. And your chromosomes are made up of a big long string of DNA, and a small portion of that DNA makes up a gene. So we've got 46 pairs of chromosomes, 23 pairs, last pair being the 22 autosomes and the last pair of the sex chromosomes. And when we talk about genetic testing, it could mean testing the chromosomes by a carrier type or a microarray, or it could mean testing single genes. So I'm just going to explain that in a little bit more detail again. So I think what's also important to be aware is that things have changed a lot in genetics in terms of what we can do from when I first started, and particularly recently in terms of the types of testing that goes on. So when we talk about old-fashioned originally, all we could really do was a carrier type, which is where you're counting the chromosomes and looking at the stripes on the chromosomes. Largely speaking, carrier type has been superseded by microarray. So microarray is where you are doing a kind of a fishing exercise for any chunks missing or extra within those chromosomes. And most chromosome testing now is done by a microarray rather than a carrier type. When you then go down onto a smaller level, bigger microscope essentially, you're then looking at Sanger sequencing, which is the old version where we had one gene, for example, a cystic fibrosis gene, where we would just look at that gene at one at a time, whereas now most testing has moved on to next generation sequencing, which is where you're looking at essentially at lots of genes all at the same time using whole genome sequencing. Just to point out that carrier types are still used sometimes. So the example you might be aware of is probably translocation Down syndrome. So if you have a pregnancy where there is an example of a Down syndrome, and it can't be you can't tell from the testing, and I'm not going to go into details at that point, but the lab will always say whether you can tell if it's a primary Down syndrome, like a standard Down syndrome, 3 copies of 21, or whether it's come from a translocation, which is much rarer but has a recurrence risk. If the lab haven't been able to tell from their report, they should recommend parental chromosomes. And for that, you do need a carrier type, because you cannot tell that from a microarray. So it is still used. But most chromosome testing is done by microarray, and most single gene testing is done by next generation sequencing now, where we can look at lots of genes all at the same time. So from a bereavement midwife point of view, and for the people who are consenting, a lot of the testing that people are referring to, I think, when they say genetic testing, is often a microarray, because that might be consented for by somebody in that situation. Most whole genome sequencing is consented for and arranged by clinical genetics still. And so when we're talking about the first line testing, you're talking about a microarray and looking at the chromosomes, whereas the next generation sequencing, that consenting is all done by us on stored samples. But hopefully that'll become more obvious as we go through. So just to show you in another way, so this is just an example of chromosome 7. If you're looking at the chromosomes in a microarray, you might see chunks missing or extra of chromosome 7. If you're looking down the microscope, they would be looking at the bands. Or if you're looking specifically at cystic fibrosis, there's a gene stored there, you would need very specific genome, genetic testing, molecular testing to look at that particular gene. Hopefully that makes sense. And I thought I'd just bring in here just way of explaining things and different resolutions, because I think we are very used to explaining genetic testing to families, but I'm not sure that everybody necessarily is. And there's lots of different ways of doing it. So when I explain to patients about genetic testing, I will often say, your genes are a bit like your instructions, and about developing maybe needs all your genes present and working with the baby to develop as you expect. And when we do a chromosome test, it's like an overview of a person's genetic makeup, looking for any chunks missing or extra. But so in some situations, we will need more detailed testing, whereby we'll need to look at each single instruction on their own and see if there's any differences in it. And so that's my kind of general spiel for how I explain things. Some people use different other explanations, and these pictorial ones are quite good. So, if you look at thinking about a karyotype, that's really low resolution. That's having your old Nokia brick phone that can just about take a picture. Going on to a microarray, a little bit more detail. The first iPhone maybe, so the picture's reasonable-ish. And then next gen sequencing is your most modern iPhone, where you can take a picture and have a really good detailed look at the picture. So some people find that sort of explanation helpful. Other people use a book analogy. I've stolen this from a patient support group, but essentially saying that, the DNA makes up the entire cookbook in this example. Your chromosomes are your chapters, and your genes are your individual recipes. So some people, it depends on who you're talking to, you might change how you explain things, but people use different ways, and it's about finding your best way that makes sense for you and the patient you're talking to.